purpose of this lab activity was to study at the same time two processes, photosynthesis and cell respiration. These are energy transfer processes by which energy is transferred from one form to another. So if we look at photosynthesis first, really quickly, you'll see that photosynthesis, first of all, only happens in plants and algae, but it is the mechanism by which energy is originally captured for almost all ecosystems. In other words, how do we get our energy? Well, technically, we're getting our energy from the food that we eat. And where did we get the food? The food typically was made by plants. So it's a very important process for capturing energy and sort of bringing it into the ecosystems originally on Earth. So photosynthesis takes carbon dioxide and water. That's the two building blocks. And then the energy from light is captured and used to basically trap the energy in the bonds of sugar. In other words, sugar is being built from the carbon dioxide and water. Oxygen is released as a waste product. That oxygen comes actually from the water. And now glucose basically contains more energy. This is sort of an endergonic reaction, if you remember. Glucose now contains energy trapped in the bonds. That glucose can then be stored, and as the energy is needed by the plant, it can then be released by breaking the glucose down. How does the plant release the energy from the glucose? By the other process, cell respiration. So in cell respiration, sugar combines with oxygen, and the sugar now is broken back down into carbon dioxide and water, and the energy that was trapped is released as ATP. And if you recall, ATP is the cell's energy currency. It can be used to run all kinds of cellular reactions, like the sodium-potassium pump, transport, movement, things like that. So these two processes are very closely related to each other. Keep in mind, cell respiration is happening in all organisms. Only bacteria are the only ones, they don't have mitochondria, so a lot of them are running an anaerobic process, a different type of cell respiration that doesn't require oxygen, but all eukaryotic organisms that have mitochondria are running cell respiration. You're running this process all the time, whether you're awake, whether you're asleep. You may run it more quickly or more times when you're running a marathon than when you're taking a nap, but this process never shuts down. Photosynthesis, on the other hand, is something that's only going to be happening in plants, and it's also only going to be happening when light is available. So technically, a plant would be running both processes, cell respiration and photosynthesis, in the daytime, but at night, they would only be running cell respiration. A plant in the dark would technically be competing with you for oxygen, because they wouldn't be running photosynthesis in the dark. They would only be running cell respiration just like you. All right, so to summarize, cell respiration is the way that we release the energy trapped in glucose. All organisms are running cell respiration all the time. Breaking down glucose, we release the carbon dioxide and water as waste products, and the energy from the glucose is captured in ATP. Photosynthesis is a process that only plants and other organisms similar to plants can do, and this is how the energy is originally captured from the sun and trapped in the bonds of glucose. The reason that we used algae in this lab, which you can see in this little picture here, this is what our algae looked like. We actually had 20 of them, these little balls of algae, and they were put in a vial. One vial was covered with foil, and the other vial was left in the light. The reason why algae work great for this experiment to study both processes is because algae do both processes. In the light, algae can do photosynthesis. The ones that would be wrapped in foil could not do photosynthesis, but they would be doing cell respiration all the time. So cell respiration would be happening in the algae all the time. Photosynthesis would only be in the ones that were unwrapped where light was available. So now that you have an understanding of why we were using algae, what were we actually looking for to happen in the algae? This is what would happen. So carbon dioxide itself, when it is in water, it reacts with water and it forms an acid, a very weak acid called carbonic acid. And this is sort of the formula for that. So carbon dioxide reacting with water, and it forms carbonic acid. There's the hydrogens showing that this is an acid. Now, notice this is a reversible reaction. The arrows go both ways. So as more and more carbon dioxide bubbles into water, the more acid would form. And if the carbon dioxide was being used or taken out of the water, then this product, the carbonic acid, would disappear. So as carbon dioxide builds up, we would form an acid. The more carbon dioxide, the more acidic the solution, and we were using an indicator. And remember, indicators change color as pH changes color. So if we had carbon dioxide building up, which, if you recall, was the product of cell respiration, would be that the carbon dioxide would build up, then the pH, which would start around here in this area, around 7.3, the pH would go down. So the more cell respiration was happening, the lower the pH would get. On the other hand, photosynthesis uses carbon dioxide. So even though cell respiration is producing carbon dioxide, if we have a bright light and a lot of photosynthesis happening, basically there would be more photosynthesis than cell respiration. In other words, more carbon dioxide needed than was being produced. So the carbon dioxide levels overall would fall. And if the carbon dioxide levels go down, going back to this original equation here, the pH would now go up. So that would mean that this hydrogen would disappear. The reaction would go this direction, and therefore the pH would go up. And so again, starting around 7.3, the ones that were in the light would go towards purple because the pH was rising because of less carbon dioxide. And that, in essence, was what we were able to do in the lab. So you took your 2 vials, one was wrapped, and you placed them under a light. And here we can see this process in motion. So here's our 2 vials. Every 5 minutes I shook the one. Technically you were actually looking at the one in the dark, but I didn't do that in this experiment. I just let it run. Notice how the one that's in the light is starting to go towards the purple range in the color. And I'll unwrap the other one at the end so you can see how its color changed. All right, so here we are at the very end. So my first one is the control. So notice this is around, give or take, 7.3, 7.5. That's where your original one would have started. This is the one in the light. You would have measured every 5 minutes, and you'll notice it was moving towards the purple range. Mine didn't quite make it there, but most of yours actually made it all the way. And this is the one in the dark. Notice it is lighter than the control, so we started here in this range, but this one ended up here. Again, reasons why that happened. This one is running only cell respiration, so the carbon dioxide levels were going up. That causes that carbon dioxide to form carbonic acid, and the pH drops. The one in the light was running both, so a little bit of carbon dioxide being made, but a lot of carbon dioxide being used for photosynthesis, so carbon dioxide levels overall went down, causing the pH to also go up, pH to go up, and it went towards the purple. So that, if you were absent, in essence, is what you should have seen happen in the lab, and you should be able to use this background information to answer any questions.